this is Professor Ikeda from Georgetown University, co-chair of this session. Next, as the next lecturer, we would like to invite Professor Ting Chen from National Institute of Biological Science, China. And today she's going to talk about mesenchymal niche-regulated regional epithelial regeneration. So, Professor Chen, please start.
Thank you. It's a great honor to be invited here, and I've learned so much so far, and I hope I can contribute a little bit. And today I'll mainly focus on mesenchymal niche, basically dermal papilla cells. And the question we're interested in our lab is one that how do cells know where they are in our body? Because so far during this meeting, I've heard a lot of talks about endogenic alopecia, and on the left you can see the older Darwin that he lost a lot of his frontal scalp hair, but his beard is totally fine. So there's a positional difference regarding about hair follicle growth and hair follicle loss. On the right is actually a very common form of depigmentation disease called vitiligo. And one of the major forms of vitiligo is called non-segmented, meaning you get symmetrical loss of melanocyte in your skin. And this is believed to be due to autoimmune attack of the melanocyte by your cytotoxic T cells. And you always see, I mean, this is a stunning picture as a model. And you can see this symmetrical localization of this loss of melanocyte. So clearly during normal development and the physiological condition, there is a positional effect of our hair follicle, our melanocyte, and even our epidermis. So the question we really know is that how do cells know where they are in our body? What's the molecular mechanism behind this zip code system? So the model system we use in our lab is mouse. And mouse hair follicles, you know, the dorsal skin and belly skin, they contain follicles that continuously cycle. They grow from quiescent stage to growth phase. And they do this throughout their lives. And we noticed that actually on the mouse ear skin, although they appear hairless, they actually do contain tiny miniaturized hair follicle. And once they're formed after morphogenesis, they do not cycle. They pretty much stay in quiescence throughout the mouse life, no matter when you check. So there's a clear distinction of whether follicles do or do not regenerate, just normally. And so we decided to use this system to ask, how do they know where they are? And the mechanism could be very complicated because just like Dr. Hoffman just showed, there are HAP cells, the dermal cells, fibroblasts, neurons, endothelial cells. There are a lot of components in the dermis that could tell the cells where they are that are all different. And luckily, there is a Mutogenesis mouse called a koala mouse line. And this is produced in the 80s due to irradiation mutagenesis, random mutagenesis study. And you can notice that on the left, on the koala mice, they have very hairy ears and muscles. And when we take a look at this koala ear skin, we notice that they are not only longer, they actually spontaneously cycles, meaning you can use a single switch to turn on the regeneration process in this location. And that gives us indication, this problem probably can be solved if it's just one switch. So what is the koala mutation? And this is a chromosome 15 chromosome inversion, meaning a big chunk, 51 megabase of the chromosome that's inverted 180 degrees. And on both breakpoints, there are no genes that has been disrupted. So it's not instantly clear which gene is actually responsible for this phenotype. And there have been many publications indicating who could be, but there lacks definitive functional study. So we decided to take a look. And because the genetic DNA doesn't tell us which gene this might be, we turn to RNA-seq of the koala isolated stem cells versus dermal cells. And I'll skip all the analysis. So basically, we didn't find a lot of useful information by the stem cell, hair follicle, stem cell RNA-seq. And instead, we found a lot of interesting genes in the dermal RNA-seq. And here's one of them. So in this 51 megabase inversion region, there is a cluster called HOX-C. And there are 9 HOX-C genes in this cluster, and 5 of them actually upregulated in a koala mutant. So on the left, you're looking at in situ where you see the red dots, meaning the mRNA signal. And on the left is actually the bottom half of the follicle in the dorsal skin. And the red part is actually dermal papilla. So HOXA8 is expressed in the dorsal skin dermal papilla, but not expressed in the wild-type ear skin dermal papilla. But on the right, you can see that in the koala ear, now this gene actually get turned off again. So this expression level correlate very nicely with whether or not a hair follicle can regenerate. Since when they express them, the hair follicle happens to be regeneration. So we want to know if there's a causal relationship. Do they actually function, to switch them on and off? And how do we do that? So on the top two lane are the genotype of the koala mice. So the koala mice are a heterozygous animal. So because the homozygous koala allele actually leads to embryonic lethality due to bone deformation. So for the koala mice, they have one wild-type allele, one koala allele that's inverted. So we argue that if the HOXA cluster is functionally responsible for this phenotype, we could delete the entire HOXA cluster only on the koala allele and leave the wild-type allele intact. And that should actually rescue the phenotype if they are responsible, right? So we use Cas9 and HGNI to specifically target this cluster. And turns out this is a striking phenotype. So on the left is the wild-type mice. In the middle is the koala. You can see the clear, hairy ear and muzzle. On the right is actually the koala strain just missing the HOXA cluster as we designed. So this is great. But what I didn't tell you in this simple diagram is that we use sort of blunt force trauma to create this deletion. Because this cluster actually contains 9 HOX genes, several microRNAs, one long-non-coding RNA called hot air, and potentially many other enhancers, right, that could regulate the genes outside this region. So even though we get this reverse phenotype, we are actually not be able to show clearly it's the gene that's responsible for this phenotype. So how do we get around that? And here's when the Cas9 technique really comes in handy. So when you use Cas9 and HGNA to create double-strand break, the repair is actually pretty random. So we had a lot of founders after we did the knockout, and this is one of the founder typically. So we designed 2 HGNA, one cut the HOXA13, one cut HOXA4, and the remaining sequence actually doesn't generate anything due to random, sequence fusion. But one of the founder, we noticed very striking. They also get HOXA cluster deletion, but the phenotype appears koala-like. And when we sequence this founder line, we notice that actually the resulting is a de novo diffusion protein that's created by the N-terminal HOXA13 and C-terminal DNA binding domain of HOXA4. So this is a phenotype. And the red is Ki67 indicating the proliferating matrix in the hair follicle. On the left is the wild-type ear. There is definitely no regeneration. And the second one is the koala. You can see robust hair growth. The third is actually the correct deletion, where we rescue the phenotype. On the last one is actually the most important for us, because this one still missed all the genes in the cluster, all the microRNAs, non-coding RNAs, and potential enhancers. What's the only thing that's remaining is actually this de novo fusion protein that's part HOXA13 and part HOXA4. So basically, you missed anything else except one HOX genes, and you still get this phenotype. So this gives us a very strong confidence that it's a HOX-C genes and not other genetic component, that's switching on this regeneration phenotype. So now we know it's a gene, we want to know where do they act. In our in situ staining, a lot of the signals are in the DP, but we also see them in fibroblasts. So we want to know, exactly where does this gene work. And to do that, we turn to a lentivirus-mediated overexpression. So basically, we use lentivirus to either overexpress individual HOX genes in the epithelium or in the dermis, and ask which one can stimulate hair growth. So let's first look at the dermal overexpression. So this is actually extremely easy experiment to do. So we use lentivirus insulin needle to inject this virus solution into P1 ear when there's just a little bump and there's no follicle yet, and all the fibroblasts are just loosely there. There is no DP. And then we wait for 21 days. And after this period, the ear follicle has finished morphogenesis. They already normally went back to quiescence at that stage, P21. So if you zoom in and look at this red signal, which represents the virus expressed protein, you can notice we targeted a lot of loose dermal fibroblasts, and we definitely target some of the DP, right? And DP is marked by AP staining in the black and white field. So you can see that, well, some of the DPs are targeted by our expression method. And this is a control, just H2BRP, nothing happens. And when we added HOXA8, something striking just happens. You notice that in this year skin, a lot of the follicles, went into regeneration stage. And what's most peculiar is that if you look closely, only hair follicles that has DP overexpression can do it. The other follicle, even just neighboring one, then when they're surrounded by fibroblasts, still overexpressing HOXA8, they do not grow. So this indicating, this protein needs to be expressed in DP to function. And so this is sort of, we still see some of the epithelial cells express, the RFP as you can see in this picture. And we want to know if this gene also functions similarly. If it just overexpressed in hair follicle stem cells, would that also, trigger hair growth? And to do that, we use, this is quantification. So, basically more than 80% of the follicles with DP overexpressed HOXA8 can go into regeneration process, and control is 0. And the second we want to do it is the hair follicle stem cell regeneration. And for this, we use the in utero injection method developed in Elaine Duke's lab. And so basically here, the epithelial cells are marked in green by K14H2 and again, the virus expressed protein is red in RFP. You can see a lot of epithelial cells, and definitely a lot of hair follicle stem cells do express them, and by quantification, none of them get into regeneration. So it's the same gene, it's almost the same, overexpression method, but they need to be in specific cell type to function, and we think they probably need some specific cofactors that's only present in DP, but not anywhere else. So now that we tied the expression of these HOXA genes to whether or not hair follicle can grow, we can go to ask a more sort of a deep molecular question, well, how do this gene express in different pattern to begin with? Why are they differently expressed in different body locations? As I showed that we now know the most important place of the action is in dermis. So we isolate dermis from the ear versus dorsal skin of the wild-type mice, and just to take a look of the epigenetic modification. So here is the RNA-seq of the expression level itself. On the first lane is the ear skin. You can see the HOXA cluster is completely quiescent. They're not expressed, as we showed by in situ. On the bottom lane is the dorsal skin, where you do see at least 5 of the HOXA genes are expressed. And if you look at the active enhancer marker, it's actually very nicely correlated with whether or not they express. That's a pretty obvious no-brainer. But if you look at the repressor, it's actually very striking. So in the ear skin, when there's no expression, the whole cluster, the region is expanding, neighboring to the or heavily decorated by this, the follicle-mediated repressive marker. But in the dorsal skin, where half of the cluster is expressed, the other half is actually very robustly silenced. And when we look at the CTCA, which mediate the chromatin looping, we can see some CTCA binding site in this junction region, indicating this cluster could be looped into 2 domains, one active, one quiescent. And I didn't put the data here. We actually took a look at the BMI1 knockout mice. BMI1 is the major component of the PRC complex. They mediate this bloom, repressive modification. And this BMI1 mice actually has very hairy hair. So this is consistent with our finding, even though not as striking as koala. So next we want to know, well, how does koala gain the expression? Because this HOXA cluster is not exactly at the breakpoint itself. It's actually pretty far away from the distal breakpoint. So we want to know how does this cluster get activated after the chromatin inversion without being touched, right, by the breakpoint. So the way we did it is just isolate the ear skin from the wild type and the koala ear, the dermal cells, and ask, what's changed? And here I want to remind you that the koala mice has 2 wild type allele and 1 koala allele. So the ChIP-seq data will be slightly mixed from the two different material. But anyway, just to get you a slight taste of what the chromatin loopings look like. So on the first is the pyramid triangle-shaped readout, or actually high-seq data from mouse embryonic stem cell that's been published. And we just highlighted this region we think indicating looping in the mouse ear skin, because we use the CTCA, the boundary protein that mediate this looping, and they correlate pretty well with the mouse ears data. So you'll notice that the HOX-C cluster is actually located within the distal PAD domain, topologically associated domain. And both breakpoints actually disrupted two PAD domains. So indicating the inversion actually can disrupt two TAD. Would that be the case? If we look at the active histone modification, you notice by the arrow that, so in this one distal TAD, there is no active enhancer modification. It's completely not there. But after the inversion, you can see a peak that's gained in the HOX-C cluster. And how could this happen? If we used HOX-C as a bait to look at what genomic region is actually in contact with this area, we'll see that in the wild-type skin, the contact area is pretty restricted to that TAD region, right, by the green peak. But if you look at the koala skin, you can see ectopic interaction of the genomic region outside the proximal breakpoint that is very rich in enhancer decoration. This de novo interaction could indicate how these HOX-C genes are now turned on. So this is all just, to most people in this room, very boring epigenetics. And I'll just call you back to your attention to one last slide that most of you will like and give you a lot of food for thought. So here, HOX-C genes, we now know that they are expressing DP, they can stimulate hair regeneration, but this is a transcription factor. And how does this transcription factor in DP can stimulate hair growth? And obviously, we did a lot of RNA-seq trying to figure out what's the downstream. And I'll save you all the trouble and just show you the key figure here. So here, we notice the Wnt signaling is dramatically changed with or without HOX expression in the ear. So we use axon 2, which is a canonical target of Wnt signaling. And here, axon 2 CRIAR will serve as an indicator. If you see green cells, meaning the cells are expressing axon 2, indicating they're responsive to Wnt. So in the wild-type ear skin, there's very little Wnt activity, very little green cells in the hair follicle or in the DP. But in the koala, we see dramatic signal in both DP and hair follicle boundary region. But if we just knock out the HOX-C cluster, and the Wnt signal is gone. So there is a direct causal relationship between the HOX-C gene expression and the Wnt response. To this point, we don't know exactly what HOX-C did to modulate Wnt signaling. We just know it's upstream. We don't know if it's direct or indirect. And we actually did a bunch of even additional experiment to show if we bypass HOX and just directly activate Wnt in the ear, we can also trigger hair growth. So this is a functional downstream as well. And finally, okay, to summarize all this data. So we think the niche expressed HOX-C genes in different body positions can regulate hair follicle regeneration landscape. And here on the top are the dorsal skin dermis, where the HOX-C clusters are divided into two domains, one active domain in pink, one quiescent domain in blue. So some of the HOX-C genes are expressing DP that allows the hair follicle to cycle and grow. But in the ear skin, the whole cluster is completely quiescent in the repressed state, and none of the genes are expressed, so they do not cycle. But if you can reverse or reactivate some of the HOX genes, either by ectopic interaction of this cluster with enhancers, or by knocking out BMI1, or in our case, by lentivirus overexpress some of them, you can trigger this regional hair follicle growth. And so this is a detailed summary of our finding. And if you don't want to remember any of this, just remember this one thing. So I'm sure most of you are very familiar with this Wellington's diagram. And Shinya Yamanaka essentially told us that this ball can move by itself uphill. They can dedifferentiate, or they can transdifferentiate if you express the correct transcription factors into this ball, which indicate epithelial cells or stem cells or whatever you like. But for most of this background, this landscape itself, which indicate the stromal cells, fibroblasts, or niche cells, they're pretty static. They do not change. They're just there. And stem cells can move around despite they do not change. And here we're showing that mesenchymal niche are not only different, they actually can be changed. They also have plasticity. So if you can figure out why they're different, then you can, essentially move them around. And that would, so basically you can move the earth to change the course of the ball, where they go. And I've heard a lot of talks in the first 2 days about how people want to treat, endogenic alopecia. And as I showed you in the first picture, the beard is, the mustache, they're totally fine. So instead of trying to use unbiased approach, try to make them grow again, if we can actually figure out what's the difference between the frontal scalp hair versus, the beard's hair follicle, then maybe there's a more natural way to switch them back. So that's the critical message I want to leave you with today. And most of the talk of the data were actually created by a grad student in my lab on the left, Yu Zhou, and she's helped by a bunch of people. And we have a lot of collaborators in China, in Japan. Actually, the koala mice, I got it from Japan. And most of the knockout mice we have in this study are generated by our collaborator in our institute. And thank you for your attention, and I'll be happy to take any questions.
Thank you very much. So now this paper is open for discussion. Please.
Mine isn't really just a question, it's more a congratulations. I thought that was an excellent piece of work as someone who's been comparing beard skin, not specifically scalp and balding skin for a lot of my career. I think it's excellent work. Congratulations. But the big take-home message, if you think of what's been a recurring question from George Cotterellis and Bob Harris and a number of people all day yesterday, was whether the mesenchyme or the epithelial level aspect is dominating from initiating. And you've very clearly shown, well, not so much initiating, but from starting a hair cycle, it's got to do with mesenchyme.
Yeah.
Of course, I support.
I used to be a hair follicle stem cell person. Now I'm sort of
a big switch. You've seen the light.
Okay.
I think that's work. I hope we can grow beard on the scalp someday in the future. So you show that hugs appear to affect the wind signaling, and there are activators and inhibitors in the ear. So did you check the BMP, the inhibitor? Is the inhibitor also affected by this mutation?
Yes, the reason I say we don't know exactly what happens, there are so many morphogens or inhibitors or ligands that's changed in our RNA-seq. So there's no way for us to do a conclusive, definition of explanation how this happens. The nice thing is that just as Howard Chen has showed before, that when we isolate the dermal cells from ear and dorsal and put them in culture, the differential express of HOX genes actually do maintain. And we're thinking the only way is probably to do a forward genetic screen. I don't know how to do that, to figure out should we bother to do that?
Okay.
Yeah, it's an intriguing question. Every time I talk about this, I got asked, how do they regulate wind? And the answer is always, we don't know yet.
Thank you.
I think this is really very impressive work. I think there's two major findings, right? HOXA8 is important to regulate.
Not just HOXA8. We tried
Right.
HOXA6 and HOXA4 that also works.
Okay,
But
so
HOXA8 is the one that's...
Several of the HOXA clusters are important to
Yeah.
activate the GP in this way.
Yeah.
On the other hand, you clarify this epigenetic mechanism of this the koala inversion, right, with this enhancer. I wonder what's the regulation then in normal hair DP, sorry, in normal ear DP, how it gets silenced? Is it a lack of activation, or is it an activation of silencing? And is it the same locus?
It's the same locus. So if we look at the quiescence epigenetic marker, it spans the whole region. And the PRC complex is sort of the hand that's putting the silencer there, but we don't know what's behind the hand. And we're also thinking, should we do another screen to find out, what's the upstream? So that we don't know either. We just know the decoration is there. And if we, lose them by DMR knockout, they do get activated, hair do grow. But we don't know who is responsible for putting that repressive mark there.
You must be burning to see whether HOXC8 or other HOXCs are downregulated and miniaturized hair follicle PP.
Oh, that's the thing. I have to confess. So now we're talking about that. Because we got a lot of input, suggestions by people saying, if you can show this happens to human hair follicle, you're golden, right? Obviously, we spend a lot of time collecting human hair follicle samples and trying to do in situ. And the HOXC genes are not expressed in the DP. Maybe our probe doesn't work. Maybe it's other HOX genes. So it doesn't look like the human follicle DP are following this exact molecular mechanism. Or could be other HOX. Because people have done microarrays and there are HOXD genes that express in DP. Yeah.
Do you think that the epigenetic switch of the HOX could be due to metabolism changes, maybe due to different normoxy between the ear and dorsal?
That we haven't thought about. But we do know that during embryonic development, the HOX genes are more expressed in distal body plan. They are less expressed in the frontal region. So we think this expression pattern could be inherited from embryonic stage. They're probably not actively, maintained or changed in postnatal life. We are looking into if they are dynamic change. We don't know if they are yet. Yeah.
Thank you for your great talk. Have you ever seen that HOXC overexpression in dermal papilla will regulate the period of anagen, or faster the growing phase, or longer the growing phase?
Yeah.
The effect of that.
We choose the most easy quantification, which is from no growth to growth. And we didn't go into the details, like how long do they stay in anagen stage, or if the follicle get bigger. And they do not appear to be bigger. And I don't think they just stay in the growth phase, because otherwise we would see like really long hair on the ear. And we didn't see that either.
So you think that the period with the same stay with the ear, not just the dorsal hair?
Normally ear skin anagen is 5 days to 10 days shorter, so that's why they appear tiny little.
Yeah.
And in its overexpression, we don't see a dramatic change. Yeah. Either very short or very long.
Even size do not change.
Size do not change.
Thank you.
We are short on the time, so
Okay.
thank you very much, Dr. Chen, for your excellent work.